recorded on my computer. Okay, so it should be recording. So we're going to go through cellular respiration today. So 6.2 is on glycolysis. And I meant to fix this, but it doesn't really matter because I know what it says underneath it pretty much. All right. So during glycolysis, if you look at the word glycolysis, glycolysis means to split sugar. There we go. And so the word glycolysis, lysis is split, glycogen is sugar. We're going to have to use a small amount of ATP in order to make ATP. So what we're showing here in this picture is that we have ADP, right? We have the two phosphates, and we're going to do what's called substrate level phosphorylation. When we do substrate level phosphorylation, and I know today is going to be like I'm speaking a foreign language, and I get that, and hopefully you guys pre-read. If not, hopefully when you go back through this, that might mean a little bit more. So our substrate here is this 4-carbon molecule, and then we have these two phosphate groups. We're going to use an enzyme here to hold on to that substrate and to transfer that phosphate, notice I'm missing one, onto ADP. This is called phosphorylation. This is using an enzyme and another. We're basically transferring a phosphate from one substance to another. Okay, so that is substrate level phosphorylation. Okay. Glycolysis, like I said, the pathway means to split sugar. And what we're going to do is this is the first metabolic pathway used. And what we're going to do is we are going to take our carbon, our 6-carbon sugar, which is glucose, and we're going to split it into 2 3-carbon molecules. This is thought to be the oldest energy harvesting metabolic pathway. Does not require oxygen. It is anaerobic. So in eukaryotic cells, this pathway occurs in the cytoplasm. So in the cytoplasm of the cell, we're going to do this specific process. There are generally kind of 3 phases to it. There's the first one we call the investment phase. We have the second, in which we're going to couple. We're going to actually, this one I like to, this is more of our redox phase. And then this here is our payoff. And so I have a blank page here because what I'm going to do is I am going to go through glycolysis with you. Right? So I'm going to draw it out because, we have that little tiny paper there and that slide. And I like to do this. What did I do with glycolysis? There it is. In a way that hopefully will make a little more sense to you. I'm just like dropping everything on my end. Okay, so with glycolysis, what I'm going to do is I'm going to start off with 1, 2, 3, 4, 5, 6 carbons. So again, this is glycolysis, split our sugar. This is happening in the cytoplasm. Does not require oxygen. No oxygen. What we're going to do is we are going to undergo an investment period where what we're going to do is we're going to take ATP and we're going to put one of those phosphates onto my sugar. Oh, I don't want that color. I hit black. 1, 2, 3, 4, 5, 6. And now we have our phosphate group there. We have a big, I call it the big oop. Looks like a big oop to me. Then we're going to repeat this. And I do have videos of me going through this. Ooh, that's going to be squished. Let me do this. I think it's easier to see on the iPad though. Now I have what I call the big poop. So this is my investment phase. I had to invest, just like any kind of business. I had to invest into this process to actually get a payoff. Now, this investment phase puts two highly energized phosphate groups on each end and causes this molecule to be very stressed out in the middle. And with that stress, what happens is then we're going to continue on over here. We're going to end up splitting our big poop into a little poo and an oop. All right. Now, what we're going to end up doing is a couple different things. We have this big old phase here, and I'm going to run out of room. So I'm going to kind of do it. Maybe I'm going to do this in one long process. There's a couple things that's going to happen. First thing that's going to happen is that we are going to put NAD plus, and we're going to make NADH. We're going to do that on both of them. All right. So that's two of them, one on each. Right. This is our redox reaction. So we pulled electrons, we reduced our sugar, we pulled electrons off of our sugar. We also are going to put some inorganic phosphate into this. And so what we get, 1, 2, 3, 1, 2, 3, we get two little poops. Okay. And then I got to continue to the next page. All right. So I had, yeah. You there?
Sorry, that was an accident.
No, that's okay. 1, 2, 3. I'm going to redraw this. 1, 2, 3, quickly. So I had my little poops. Maybe I had a question. All right. So what we're going to do here is we are going to take, again, that substrate level phosphorylation. Just going to abbreviate phos. Okay. And we're going to take, we're going to do it twice. We're going to take our ADP and we're going to pull a phosphate group off. And notice how we have 1, 2 phosphates. And so I'm going to make 2 ATPs here. Okay. And then I'm going to end up with just this pyruvate. And then I'm going to do the same thing on the other side. All right. So I'm going to put in 2 ADPs and get out 2 ATPs. And get another pyruvate. All right, so to summarize this, and I'm going to wait because some of you guys might be drawing this, let you get caught up a little. All right, to summarize, so I put in, and when I got out, I put in glucose, and I got 2 pyruvates out. I'm gonna run out of space there. I put in 2 ATPs and I got 4 ATPs. So right here, my profit only is 2 ATPs. I also put in 2 NAD pluses and I got out 2 NADHs. So basically, I didn't get a whole lot of energy. In fact, we know that for 1 glucose, I'm going to get approximately, anyway, it's a range depending on your textbook. This textbook, the new OER that we're using, she uses the number 32. I like 36 to 38. Again, some textbooks go into that. I'll explain where the 36 and 38 come from. The 32 is more of a realistic ideal phase because when we do 36 to 38, we're assuming we're not losing any energy to entropy or heat, which we know is not true. Okay, so that is glycolysis. So here is where she goes into her inputs and outputs. So we put in the glucose, we got out the 2 pyruvate. Like I said, we have the 2 ATPs. We put in, that was our investment. This is what we got out, right? We put in that and we ended up with 4. So our profit was only, because we had put in 2 here, our profit was only 2. We put in 2 NAD pluses. Remember, those are our electron taxi cabs. They pick up those hydrogens, which carry protons and electrons. So basically this whole time, we're going to be reducing the sugar, the glucose. Remember our oil rig. Oxidation is lost, reduction is gain. Okay. Ooh, sorry. We're oxidizing the sugar. See, I shouldn't have taken that nap before class. Or I should have done my little oil rig in my head. This is being reduced. We're reducing here. Remember when I said you can't have one without the other? We're reducing our NAD plus. We're oxidizing the sugar. All right. So this is a complete oxidation process of pulling off electrons off of sugar. We're harvesting those electrons. We haven't done a whole lot in glycolysis. We have a little YouTube video here, which it's okay. I'm going to show it. It's just a couple minutes long. As soon as I get past the advertisement here.
My job is to make college easier because students have a lot of fun. Sorry, Coach.
Come on, skip.
ATP is the key energy molecule for all biological organisms. One source of ATP is glycolysis, a series of reactions that break down sugars while producing ATP. Glycolysis is a series of 10 reactions that converts sugars, like glucose, into 3 carbon molecules called pyruvate. Glycolysis is an important pathway for cells because it produces ATP and other products such as NADH and pyruvate that can be used later to produce more ATP for a cell. This key process takes place in the cytosol of the cell. Let's take a look at an overview of the key steps of glycolysis and focus on the input and output molecules. The first 5 steps of glycolysis convert a 6-carbon sugar, glucose, into 2 3-carbon sugars, glyceraldehyde 3-phosphate. To do so, ATP is consumed in the first and third steps. Both of these chemical reactions are catalyzed by an enzyme known as a kinase, and both reactions are irreversible. The next 5 steps of glycolysis produce energy. 1 ATP is made per glyceraldehyde 3-phosphate in both reactions 7 and 10. This means a total of 4 ATPs are produced in glycolysis. Both of these steps are carried out by a kinase reaction. Note that step 7 is reversible, while step 10 is not. It is important to remember that the products in steps 6 through 10 are formed for each of the glyceraldehyde 3-phosphate molecules generated in steps 4 and 5. This means that a total of 2 NADH and 4 ATPs are made in glycolysis. Because 2 ATPs were spent in the early steps of the pathway, glycolysis results in a net of 2 ATPs. There is one more key product of glycolysis to consider, called NADH. NADH is formed in the 6th reaction by an enzyme called a dehydrogenase. NADH is an electron carrier with the potential to produce more ATPs through a process called oxidative phosphorylation. Details of that process are shown in a separate animation. As you can see, glycolysis is a process involving multiple enzymatic reactions that break down sugars. Its major contribution to biological organisms is the production of ATP, which is used to drive many other biochemical pathways.
Okay, so that's kind of an overview of what we just watched. All right, the keynote. All right, so moving on to 6.3, the citric acid cycle. So with the citric acid cycle, like I said, we just made two pyruvates. You got to remember that one glucose gives us 2 pyruvates. That's the 3. Whoop, that's the 3 carbons, right? It is now going to undergo what we call pyruvate oxidation. Pyruvate oxidation has so many different names. It's often called I call it the intermediate step. It's been called that for the longest. And then the new book that we used to use called it the preparatory step. And I'm guessing she's calling it oxidation, pyruvate oxidation now. And so we have to undergo this step before we go into the Krebs cycle. And so what happens is we're going to remove this up here. And if you look at that up there, that is CO2. And so a lot of you guys are maybe wondering, well, where does the CO2 come from? Well, this is one of the places the CO2 comes from that you're exhaling. And then we're also going to do what? We're going to reduce our NADH. We're going to strip off more electrons. And then we're going to attach right here. This is an enzyme. If I could do that. Oh yeah, the white shows up. That's an enzyme. This enzyme is going to speed this process up. And then when we hook up to our remaining 2 carbons, right? We lost 1 carbon. So we have 2 carbons here. And then our enzyme is going to hook up, and this is called acetyl-CoA. Acetyl-CoA will then go into the Krebs cycle. So here's another picture of that. So notice we have our 3 carbons, right? My black circles I was drawing earlier. Pyruvate. We're going to lose 1 to CO2. Coenzyme A comes in. We're going to pick up some electrons right here, picked up electrons. We have our acetyl-CoA, and that is going to get fed into the citric acid cycle, which is, this one has a couple different names. I'll explain why it's called the citric acid cycle in a minute, but it was called that first. And then the man who came up with all the steps, Dr. Krebs, is why we call it the Krebs cycle now. So if we talk about, I don't know if I want to, I'm going to do the Krebs cycle first here. Do I want to do the Krebs cycle? I'm trying to decide whether, no, I'm going to do intermediate step. So intermediate step. We have our 3 carbons. Now, remember, we have 2 of them, right? I don't want you forgetting we have 2 of them because remember that glucose was split into 2 pyruvates. You may also see this is called pyruvic acid. There's 2 naming systems we use, old organic naming system and the new one. And then we're going to end up losing a molecule of CO2. We are also going to end up getting NAD coming in and being reduced, picking up more electrons. And then we're going to have our enzyme, our coenzyme A come in. And then we're going to end up with acetyl CoA. And again, this is happening now. What actually, I don't know if she talked about this. I'm going to talk about this. This happens we're inside the mitochondria now. So where glycolysis occurred in the cytoplasm, pyruvate has to go into the mitochondria. And we're actually inside in the matrix, in the very interior of the mitochondria. So that pyruvate gets transported into the matrix of the mitochondria from the cytoplasm. And then we're going to go into the citric acid cycle. So we can see here's our acetyl-CoA. Now we have 2 carbon molecules, and it's going to hook up with 4 carbon molecules called oxaloacetate. And so when we have 4 carbons plus 2 carbons, we're going to end up with 6 carbons, and that is citric acid or citrate, which is where we get that citric acid cycle from. Next, we're going to notice we lose a CO2 and we pick up electrons, and now we have 5 carbons. We're going to repeat that, lose CO2, have our reduction, pick up more electrons. Now we're down to 4 carbons. We are also going to have enough energy between these steps here to make ATP. We are also going to pull off more. We have another reduction here with a molecule called FAD. That is another type of electron carrier. And so with that electron carrier, it is going to pull off and take electrons. We're going to see where it goes next. And then we have one more reduction here with our NADH. And then we're back to oxaloacetate. So step 1, the acetyl-CoA comes in, it escorts that 2 carbons in so it can hook up with the 4 to give us 6. And then we're going to have a series of, like I said, those redox reactions. We're oxidizing our sugar, we're reducing our electron carriers, we're releasing the CO2, again, more CO2, where we are exhaling it. Remember, pyruvate had 3 carbons. 1, we lost the CO2 in intermediate step. The other 2 we lost in the citric acid cycle. And that's what's happening with the sugar. You're burning the sugar off. We're getting rid of the carbon and oxygen, and we're taking that hydrogen. And that is what we're going to use to fuel our process of making ATP. All right, so I'm going to go ahead and draw for you guys. My pen keeps timing out. My citric acid cycle. So I have 1, 2, 3, 4. That is my oxaloacetate. Four carbons. It is going to hook up with my 2 carbons in acetyl-CoA to give us 6 carbons. The citric acid. Okay. Now we're going next step. We're going to go next step. Oh my gosh, I didn't lose it. Nope. Next step. 1, 2, 3, 4, 5. We lose a molecule of CO2. Lost a carbon. In that process, not only do we lose a carbon, we are also going to pick up electrons. We're going to repeat. I'm going to make this a little longer because we have a couple things happening here. Now we have 4 carbons. Lost a carbon. We also are going to make a molecule ATP. We have enough energy between the 5 and 4 carbon molecule to generate enough energy to do substrate level phosphorylation here. So we have another ATP. And then we're going to go here. There's a lot of steps going on here that you are not seeing. We're going to take our FAD. We're going to load that up with hydrogens. And then we're going to go one more step here. We're going to pull off some more electrons. All right, so now we have all these taxicabs, all these electron taxicabs, these electron carriers are all full. Where do they go? It's like being, at a concert and trying to get a ride home. You're trying to taxi, trying to Lyft, trying to Uber, everybody's full. Everything backs up. They got to be able to take their customers and drop them off somewhere so they can empty it up and come back again. Because I'll tell you right now, if you don't have any more NAD pluses that are empty, you can't go to the next step. You're stopped at citric acid. So we have to have a place to dump all of these electrons off. Plus, we still only made 4 ATPs, right? Because we have 2 of our acetyl-CoAs, so that we have to go around twice, right? Once for each one. That's 2 ATPs there. We made 2 ATPs in glycolysis. That's only 4 ATPs. That's not much energy. All right, so when we combine these stages, like we said, we have 4 ATPs, 2 from glycolysis, 2 from citric acid. We have all of our carbon dioxide that we're exhaling. We have made 10 NADHs. Remember, we made 2 in glycolysis, and we had 2, 4, 6. Remember, we had 2, 4, 6 because we had to double it in citric acid cycle, and we had our 2 FADH2s. All right, we have now pulled all the electrons we can off that carbon. Those carbons are useless. They are being exhaled. So that brings us to the end of 6.3, and now we're going to move to oxidative phosphorylation. And just like the word implies, we are going to phosphorylate using oxygen. We talked about where all the CO2 comes from that you exhale, but I have not yet talked about why we inhale oxygen. And I ask you, it's like, why do you need oxygen to live? And a lot of people say, oh, I'm like, well, why do you need oxygen? And people say, oh, to live. Well, why do you need it to live? And they will go around and around all these different things. Oh, it needs to go in your blood. Why? Well, your tissues need it. Why? I'm going to explain to you right now why you need oxygen. So we have 2 parts in oxidative phosphorylation. We have what's called the electron transport chain, or ETC. We have a second part called chemiosmosis. Now, so far, we have been in here in the matrix in the middle of the mitochondria. Remember, the mitochondria has an inner and an outer membrane. So we have the outer membrane, and then we have this inner membrane that's convoluted, and then we have the space in between called the intermembrane space. We are going to be working now within this inner membrane. All right, so inside that inner membrane, we have what's called the electron transport chain. Remember, our NADH here is going to drop off its passengers. And as it drops off its passengers, it's going to do work. And it's going to pass and pass and pass and pass. And as it passes through, these proteins here are called proton pumps. And for every set of electrons that go through, it's going to pump a hydrogen, an H+, a proton. So we're passing, 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 passing. The problem is when we get to the end of the chain here, who takes the electron? The electron's got to go somewhere. Electron can't just disappear. It's got to go somewhere. If we do not have a place to put the electron, everything backs up and this is full and we can't dump it off. We're back to our problem. Okay. So what happens is oxygen, remember we said oxygen is highly electronegative. Oxygen says, I will take the electron. Okay. And when it takes the electron, it can take the electron and it can combine it with a proton. And an electron and a proton gives us hydrogen. And that gives us metabolic water. So oxygen, I'm going to drive my point home. Oxygen is the final electron acceptor in ETC. It's the final electron acceptor. Now, remember in Krebs cycle, I said we had a different flavor or different color taxicab. That different color or flavor taxicab is FADH2. It also picked up electrons in the Krebs cycle. It has a different drop-off location. It's going to drop off here, but it's going to do the same thing. It's going to drop off the electron, pump, pump, and then that electron is going to go back onto oxygen. So we have two different flavors. Actually, NAD is based off of niacin, and which is a vitamin, and FAD is based off of the flavonoids. They're all B vitamins. Now, as we are pumping those protons into that intermembrane space, our proton concentration is getting high. It's high on this side of the membrane. It is low on this side of the membrane. We have now established a proton concentration gradient. We did work. We actively pumped. This is where we're using all those terms from chapter 5. We did active transport, but instead of using ATP, we use those energized electrons we dumped off to drive those pumps. And now we know that by passive transport, the protons want to diffuse back inside where it's low in concentration. They want to go downhill. And this protein here is called ATP synthase. Now, ATP synthase it actually turns. It'll actually turn, and we'll see it in the little video, and it will provide enough energy as those protons are going through it to put on a phosphate group onto ADP to make our ATP. And again, so we're doing this is with oxygen. We'll talk about what happens next week without oxygen. All right, so let me go ahead and draw this out for you. So here is our inner membrane. So this would be the inner membrane. This is the intermembrane space. We could say this would be the outer membrane. We're going to have 3 proton pumps. There's a proton pump. Since I had poops earlier, I might also have PPs here. PP, proton pumps. All right. We're going to first drop off our NADH. Now we're oxidizing it, right? Oxidation is lost. The proton pump's being reduced. NAD+. We're dropping off our electron. Now that's a highly energized electron. And when I do that, it is going to pump, what color do I want to use? Protons, pump to proton. All right, I have a couple more proteins in my inner membrane. We're gonna pump. Okay. Notice I went down in energy. As I did work, and what work am I doing? I'm pumping a proton. I got to put another protein in there. Pass the electron, pass the electron. Pump another, proton. The proton concentration up here is getting high. Down here, it's getting low. And I'm at the end of the road. Oh, what am I going to do now? Well, I'm going to tell you what I'm going to do now. I'm going to take oxygen and I'm going to combine that with any of the protons that didn't get over there. And I'm going to make water. That is my final electron acceptor in ETC. All right. Now remember, I have my second taxi cab here, my FADH2. I'm going to reduce that or oxidize it, right? Oxidation is loss, reduction is gain. So I'm going to drop off those electrons. They're going to do some work too. And now that these are empty, right, they can go back to glycolysis, intermediate step, Krebs cycle, wherever they're needed. They can go back, pick up more electrons, return to ETC. Because remember, all of this right here, that's ETC, electron transport chain. And then we're going to have our final piece to our puzzle right here, our ATP synthase, which is what we have been building all of this up for. And as those protons diffuse down their concentration gradient, we are going to make ATP. Whew. And this part right here, this part right here is called chemiosmosis. The whole thing is called oxidative phosphorylation. Okay, I'll wait here in case you guys want to finish copying it. Or some of you guys might have taken screenshots. I don't know. There's all kinds of different things. I definitely, for practice, would draw it out a few times. I would draw it out and I would say it out loud as you're doing it. NAD drops off at the first proton pump, passes electron, pumps proton, passes electron, pumps proton, passes electron, pumps proton, then goes on oxygen. Oxygen combines with protons to make water. Proton concentration drives ATP synthase, and then we put a phosphate onto ADP to make ATP. So I would be doing this a couple times. So an overview here, the electron transport chain is a series of electron carriers embedded in that inner mitochondrial membrane. Notice we're going to oxidize NADH and FADH2 and shuttle electrons to oxygen. Protons are pumped from the mitochondrial matrix into the intermembrane space to make that concentration gradient. Oxygen is the final electron acceptor, takes the electrons, the protons, reducing it to make metabolic water. Each component of the electron transport chain is more electronegative. Talked about electronegativity. All right. This really is not a game of friendly pass the electron. In reality, what happens here is that oxygen is a big bully and takes the electron from this last proton pump. This last proton pump kind of goes like, hey, wait a minute, and knows it would get its butt kicked by oxygen, but it could take it from cytochrome C. And then cytochrome C is like, really? And then cytochrome C takes it from this proton pump. And then we've got, I always get this, I want to call it coenzyme Q. And then we go here, and then we just keep stealing it. So finally we get to NADH, and then it steals it from NADH, but that's okay because NADH wants to get rid of it anyways, because he wants to empty up his cab and go get some more from one of the other metabolic processes. So this is a series of redox reactions, and it's based on electronegativities. This is all chemistry. So you may want to know, well, why do you need to know so much chemistry for biology? And this is it. There is a lot of chemistry in biology. And so potential energy is then used to pump hydrogen ions. So as we're moving those electrons, that energy creates that potential energy, which gets converted to kinetic energy, and we build that hydrogen ion gradient, which can be used in chemiosmosis. And like we said, at the end of ETC, oxygen's going to take it, right, and then reduces it to make water. Chemiosmosis uses the kinetic energy, that's that energy of moving those protons from high to low, to drive that protein to make ATP. Here's a picture of it all in. I like this diagram because it's showing you the citric acid cycle in the matrix. We'd also have the preparatory or intermediate step in here. Right out here, we would have in the cytoplasm, we would have glycolysis. And then pyruvate would go into the preparatory step would go into the citric acid cycle. And then all of those products that we made in those different cycles would go into the ETC, which we would pass the electron, make that concentration gradient, drive those protons back through, and drive ATP synthase. And so we have another little video here that's going to show you that ATP synthase. Which the one thing I do like about the video, come on, is that it shows you how it kind of turns.
The electron transport chain is a series of protein complexes embedded in the mitochondrial membrane. Electrons captured from donor molecules are transferred through these complexes. Coupled with this transfer is the pumping of hydrogen ions. This pumping generates the gradient used by the ATP synthase complex to synthesize ATP. The following complexes are found in the electron transport chain: NADH dehydrogenase, cytochrome BC1, cytochrome oxidase, and the complex that makes ATP, ATP synthase. In addition to these complexes, two mobile carriers are also involved: ubiquinone and cytochrome C. Other key components in this process are NADH and the electrons from it, hydrogen ions, molecular oxygen, water, and ADP and PI, which combine to form ATP. At the start of the electron transport chain, two electrons are passed from NADH into the NADH dehydrogenase complex. Coupled with this transfer is the pumping of one hydrogen ion for each electron. Next, the two electrons are transferred to ubiquinone. Ubiquinone is called a mobile transfer molecule because it moves the electrons to the cytochrome BC1 complex. Each electron is then passed from the cytochrome BC1 complex to cytochrome C. Cytochrome C accepts each electron one at a time. One hydrogen ion is pumped through the complex as each electron is transferred. The next major step occurs in the cytochrome oxidase complex. This step requires 4 electrons. These 4 electrons interact with a molecular oxygen molecule and 8 hydrogen ions. The 4 electrons, 4 of the hydrogen ions, and the molecular oxygen are used to form 2 water molecules. The other 4 hydrogen ions are pumped across the membrane. This series of hydrogen pumping steps creates a gradient. The potential energy in this gradient is used by ATP synthase to make ATP from ADP and inorganic phosphate. The ATP synthesis steps you see here are discussed in greater detail in the ATP synthase gradients animation. This animation illustrates 2 full cycles of electron donation. In biological systems, however, many electron transport cycles occur simultaneously, helping to ensure that the proton gradient is always maintained.
Like I said, I like how they show the ATP synthase does move, that it turns. All right, so what I want to do here is I want to summarize what we have done. All right, so the first step we did was glycolysis. And in glycolysis, we got out 2, remember we profited 2 ATPs, and we made 2 NADHs. And then we had the intermediate step. Or preparatory step. And then there we made 2 NADHs. We only made 1, right, in the process, but we had 2 pyruvates. And then we had the, what color do I want to use? I'll go back and forth. We had the Krebs cycle. And in the Krebs cycle, we made 2 ATPs, 6 NADHs, and 2 FADH2s. Right. So if we look at this, so far, all we've made is 4 ATPs, 10 NADHs, and 2 FADH2. We still don't have anywhere near our 30s, but for every pump, hopefully this stays when I move this. When I go through a pump, every pump that I go through pumps 1 proton, and I get 1 ATP for every proton. So for 1 electron that NAD drops off, I get 1, 2, 3 protons, which will drive, yes, it's still there, 3, it'll make 3 ATPs. So that gives me 30 ATPs. Now remember, FAD, let me go to a different picture. FAD only goes through 1, 2 pumps. So I only get 2 ATPs out of FADH2. All right, so I only get 2. So that gives me 4 ATPs. So if I look at this, 4, 30, and 4, that is 38 ATPs from 1 molecule of glucose. Now, remember how I said that pyruvate is made in the cytoplasm and it has to get into the mitochondria. It gets in through an active transporter. The active transporter requires energy. And so there is a fee of 1 ATP per pyruvate. So we're going to lose 1 ATP per pyruvate. And I had 2 pyruvates. So that means I'm going to lose 2. All right. So that means I have a total of 36 ATPs. But like I said, that's in a perfect system. All right. On your test, I am not going to ask you How many ATPs? Well, I am going to ask you how many ATPs do you get from a molecule of glucose, but my question is not going to be 32, 34, 36, 38. It'll be something really different, like 32. No, I don't even do 32. I'll do like 10, 4, 36, 92 or something like that. So it'll be really, far numbers. So look at your low to mid 30s. That would be what you're looking for. All right. So and when we do our net here, we get a net of 2 ATPs from glycolysis, a net of 2. Remember, we made 2 in the citric acid cycle, and 28 molecules of ATP during oxidative phosphorylation. Right. So we made 10 Na. Remember, we talked about that. If we look at this here, we have 6. Okay. We have 6 NADHs for every, because we had 2 pyruvates, and that gives me 6 times, we talk about over here, 6 times 3. And so that would be 18. So she's kind of split them up. I like to add them all up. So she just goes the NADHs there and then puts them here and then puts them here. But her number is approximately, so a net of 28 molecules of ATP for oxidative phosphorylation, 25 from, we talk about the oxidation of NADH, 3 molecules of ATP from oxidation of FADH2. So when she gives you your total, come on, here is her total down here, which is approximately 32. Like I said, in your OER, she's going to come up with 32, right? So do not focus on the identical numbers. Do not focus on step by step. What I want you to focus on is the process. What is happening in glycolysis? What is happening in intermediate step? What is happening in citric acid cycle? What are you oxidizing? What are you reducing? What's happening to the oxygen? Why do we need oxygen? Where is the CO2 coming from? So again, I'm not asking for, and if you go online and actually Google glycolysis, you would be like, holy crap, there are so many steps and enzymes and water molecules going in and out. There are so many more steps than we talked about. But what we want you in this class to do is just understand the concept of these processes. Okay. So I am going to, at this point, I'm going to